In this video is X inactivation and how it contributes to the formation of bar bodies. X inactivation, which is sometimes called lionization after the woman who first discovered it, is where one of the two X chromosomes in a female is shut down and compressed into a bar body, which is a dense clump of mostly inactive genes. And this is done because 1 X worth of genetic information is the normal amount or dosage for humans, which is why men can actually survive with only 1 X to begin with, because we only need 1 X, and Y mostly codes for male sex chromosomes. Now, this inactivation process is random from cell to cell within the female. So some cells will shut down the paternal X, and some will shut down the maternal X, which leads to a sort of genetic mosaicism, almost. And a good example of this is the tortoiseshell cat, which is almost always a female. The random inactivation of the X from one parent makes patches of tan or orange fur, and the random inactivation of the X from the other parent makes patches of black fur, and there's no real pattern to it because it's selected just randomly from one cell to the next. And there's always one less bar body in a cell than there are X chromosomes, meaning that the average female will have 1 bar body, and the average male will have no bar bodies at all. However, these numbers can be different in people with sex chromosome aneuploidy. Men with Klinefelter syndrome, for instance, have 2 X chromosomes instead of 1, giving them an XXY genotype, and as a result, they have an active X, a Y chromosome, and 1 bar body in each cell, whereas women with triple X syndrome have 3 X chromosomes instead of 2, giving them an XXX genotype instead of XX. But instead of having 2 active X chromosomes and 1 bar body, they have 1 active X chromosome and 2 bar bodies in each cell. And this is interesting because it indicates that actually being inactive is the default status of the X chromosome in females, but one of them is selected to become active rather than the other way around, which is where they both start out active and one is selected to become inactive. And there can be spontaneous reactivation of the bar body X. A 2008 study indicated that breast cancer patients were statistically significantly more likely to have a reactivated X chromosome than controls, although why is unknown, and there is evidence that physiological stress can lead to the reactivation of the bar body X. It's important to note that not every gene in the bar body X is inactive. Most of them are, but not all of them. Anywhere from 3 to 15% of the genes are still transcribed, and the presence or absence of these genes and how they interact with the pseudoautosomal regions is thought to be what causes the symptoms in individuals with sex chromosome aneuploidies, like Klinefelter's and Turner syndrome, which is where a woman has what's called an X0 genotype, meaning that she lacks a second X chromosome entirely, and so she lacks some of those genes that she would otherwise have. In the lab, bar body X's can be used to determine whether a type of cancer is monoclonal, meaning it arises from just one cell, or whether it is polyclonal, meaning that it arises from multiple cells, which can indicate how you should treat it, and shows that bar bodies are much more than just sex indicators, And they do, in fact, have a good deal of research potential, particularly in the disease origin